2 Wednesdays ago?
Yeah, 15, I think different chapter. We don't cover that chapter.
Oh, okay. I
Yeah.
wasn't sure because it's on the study guide.
Is it? It is not in the study guide, I believe.
Oh, well, I didn't.
Yeah. Like we have covered the central nervous system, autonomic nervous system, peripheral nervous system, and then after that we have a spatial senses. So
Yeah, chapter 15 is the autonomic nervous system.
we covered that.
Oh, we did? Okay, so that
Yeah,
was condensed into chapter 14 and 15?
yeah. Chapter 14, 14, we have touched only very little. That's the peripheral nervous system, and I have put them together.
Okay, okay.
Yeah,
Thank you for clarifying.
thank you. Okay, guys, welcome back after a week of restful spring break. And this week is we're going to talk about spatial sense. And after that, we have another midterm. So in spatial sense, organs are the eye, ear, nose, and tongue. We'll focus mainly on eye and the ear, and we will touch the base with taste and smell. Okay. And today we will focus on the eyes. So the chapter outline for chapter 16, the spatial senses, we will go over what is senses, and senses are two types: general senses and spatial senses. And in spatial senses, there are some spatial names like taste and smell are chemical sense because our body detects chemical compound. That's why we call them chemical sense too. And then we will go over the eye and vision. In the eye, we will talk about accessory structure of the eye, which is outside of your eyeball, like eyelashes, eyelid, eyebrows, those structures. And then we'll talk about the eyeball and the anatomy of the eyeball. So we will go over gross and microscopic structure of anatomy. And most importantly, we will talk about the tunics, the layers of the eyeball. Okay. And then we will go over visual pathway. In next class on Wednesday, we will go over the ear. And the function of ear is hearing and equilibrium, which is also called the balance. We'll go over external ear, middle ear, and then internal ear. Internal ear is also called the labyrinth. And similarly, we will talk about the auditory pathway. Okay. So the sense is the perception to external stimuli. Like when there is light and your body respond to light, that is light perception. When there is heat outside and your body respond to it, that is response to stimuli, external stimuli, heat. When you have a bad food inside your tummy and your tummy irritates and you try to throw up, that is the stimuli. If you have increased sodium ions in your plasma and you feel thirsty, that is a stimuli. So these senses are perception to external stimuli, and stimuli can be anything. It can be physical, chemical, biological, microbiological, anything. It can be a stimuli. Okay. So our body response to external stimuli is sense, and sense are two types: general sense and a spatial sense. General means general sense means it can be perceived by pretty much all part of the body. So let's see touch, whether you touch your eye or your leg or your hand or your nose, each part of the body can perceive to the external stimuli, which is touch, which is the deformation of plasma membrane of your cell, and you perceive it. So if you can perceive a sensation from all part of the body in generalized body part, then we call it general sensation. And if there is spatial organ to perceive a particular stimulus, then we call them spatial senses. And but by the way, spatial senses organs are all congregated in your head region. They all are there, like your eye, ear, nose, tongue. They all are in the head region. So spatial senses are the vision, hearing and balance, taste, and smell. And the organs are eye, ear, nose, and tongue. Okay. With that background information, we're going to talk about now the first part of the spatial senses, which is taste, smell, and vision. Taste and smell, we're not going to spend time, okay? Taste, smell, sight, hearing, and balance are spatial senses. Touch is a general senses, and this is a large group of general senses because touch receptors are found pretty much everywhere in your body. Spatial sense receptors are not like touch receptor. Usually touch, pain, pressure, vibration, those receptors are free nerve endings, whereas spatial sensory receptors are localized. They are confined to the head region of our body. And receptors are not free nerve endings like general senses. They are spatial neuron or spatial receptor cells or modified epithelial cells. So they can be several kinds. So the spatial sensory receptors are specialized receptor cells, modified, sometimes modified neurons, or modified epithelial cells. Okay, so this is the spatial senses, different from general senses. Okay, taste is another term for taste is gustation, and for the smell is olfaction. And receptor for taste and smell are chemoreceptors because these receptors detect chemical compounds. So for example, when I put salt on my tongue, the salt get inside the receptor and then receptor is depolarized. So tongue receptors are detecting the chemical compound, particularly sodium ions inside the salt, sugar molecules inside the sugar. That's why we call them chemical receptors or chemoreceptors. Eye and vision, similarly your tongue too on the taste, and similarly they smell. Like when you sniff something, the chemical compound get inside the receptor and that produce action potential which reaches to your temporal lobe of your brain and you smell that particular odorant molecules. Okay, so now eye and vision, the organ for eye is your organ for vision is your eye. Okay, that's why we call them visual organ, and it is highly sensitive organ. Eye is very sensitive organ. 70% of all sensory receptors in the body are in the eye. So you can see how many receptors are squeezed in that small eye. 40% of cerebral cortex is involved in processing visual information. Occipital lobe, like primary visual association area, primary visual cortex, visual association area, frontal cortex, frontal eye field, several regions of the brain are involved in processing the vision, the light. When you see somebody's eyes, you see only 1/6 of the surface of eye. Otherwise, 5 part of those 6 part of the eye are inside the cavity in your face, and that is called orbital cavity. So most of the eye are fit inside the orbital cavity. Only 1/6 you can see. Okay. So now we're going to talk about eye, the structure which we can see from outside, gross anatomy, but external structure. That is called accessory structure of the eye. You don't need these structure to see things, but these structure protect, help your eye to see. Okay. So those structures of the eye are called accessory structure of the eye, and they are eyebrows, eyelid, and inside the eyelid you have lacrimal carbuncle, tarsal plates, and tarsal gland. So let's see what are they. Eyebrows, everybody knows that is the coarse hairs on the superciliary arches. So when you see superciliary arches, your upper eyelid, do you remember the frontal bone? We said there is supraorbital ridge, supraorbital margin. So this ridge, supraorbital ridge, is the on top of that ridge you have eyebrows. Okay, eyelid covers your eye, and another terminology for eyelids is palpebri, palpebra and palpebri. Palpebra is singular, palpebri is plural. So eyelids separated by palpebral fissure, because between two eyes lid there is a tear, like cut opening, and that opening is called the fissure. And these two fish eyelid, both side of the eyelid, meets medially and laterally, and those medial point is called canthus, singular, and plural is canthi. And the radius elevation at the medial canthus is called lacrimal carbuncle. And on the bottom, inside of the eyelid, you have connective tissue uh, lining, and that is called tarsal plates. And inside that lining, we have glands, that is modified sebaceous gland. So when you have sometime infection or the allergic reaction of the eye, and you see a lot of white mucus uh, build up in the medial canthus, that is coming from these tarsal glands. And these glands get infection with the when you have red eye. So here you go. This is the subject's right eye, and this is the uh, sagittal section of the eye, subject's eye. Okay, so right side of the right side of the eye is removed, and you can see the section of the eye. This is frontal view. This is posterior view. Okay, so you have cut half an eye, and interior part is removed. So in the accessory eye structure, here you go. This is the eyebrows here on the superciliary, and then here is eyelid. This is the superior eyelid, inferior eyelid, and the opening is the palpebral fissure, because another name for eyelid is palpebral. Inside the eyelid, you see there is tarsal plate. This is the connective tissue plate, and on the connective tissue plate there is tarsal glands here under the eyelid, and then there is eyelashes on the lid. These eyelid and eyelashes protect foreign particles from getting inside your eye. The levator palpebris superioris muscles help in opening your eyes, and there is another muscles in the eye, that is orbicularis oculi muscles, which also help in closing here. The eye has thin membrane covering. The eye is covered by thin membrane, and that is called conjunctiva. So this layer, you see flapping layer, this layer is called conjunctiva. This conjunctiva has two layers. I want you to pay attention because people get confused here. So eye has the cornea anteriorly here. We're going to go over that later. And there is dark part, and there is pupil. So the whitest part of the eye is covered by a thin membrane, and that membrane is called conjunctiva. So conjunctiva has two layers. The thin membrane, which is covering the white layer of the eyeball, because it covering the bulb of the eye. That's why we call it bulbar conjunctiva. But the same conjunctiva then flip back and also covers inner side of the eyelid. You see this area. And that conjunctiva, this one, is called palpebral conjunctiva. So this is palpebral conjunctiva and this is bulbar conjunctiva. And between these two conjunctiva, there is a cavity, a space, and that is called conjunctival sac. And this sac is important clinically. When you put, like when you retract your eye and then we put drops in your eye, your drops first get into this cavity and hold here. Or when you put eye ointment, they come here. And from this sac, then slowly it mix and goes all over through the tear. Okay, so that is called conjunctival sac. This is the accessory structure of the eye. Okay. Another accessory structure of the eye is the lacrimal gland. Lacrimal gland, another name for lacrimal gland is tear gland or tear producing gland. This gland is lying under your upper eyelid in the lateral part of your orbital cavity. So if I put my eyes here on the right side here is under my eyelid. Here is the lacrimal gland. Lacrimal gland has a lot of blood vessels and it produces watery secretion that is called tear. And then tear through this small duct, excretory ducts of the lacrimal gland, drain into your eyes from the lateral side. And then it bathe your eyes because eyes is covered by epithelial tissue. And you know epithelial tissue cannot leave dry. So it needs to be moist to protect from atmospheric dirt, atmospheric microorganism, atmospheric temperature. And after bathing and cleaning the eye, these tear then come in the medial side, in the medial canthi. In the medial canthi, you see there is two small ducts opening. That is called lacrimal puncta. They enter through that lacrimal puncta and then get inside the two lacrimal canaliculi, superior and inferior. Lacrimal canaliculi then joins the large lacrimal sac, drain into nasolacrimal duct, and then drain into your nasopharynx, which is pharynx behind your nose. And then your tear get inside your tummy, in your stomach, through the throat. That's why when you put some like eye drops in your eye, and if their tests are bitter, you can taste sometime. Or when you are emotional, you're crying and you are producing a lot of tear. In that case, these two ducts cannot handle the production of tear, and then tear drops down here outside. Okay, this is called the lacrimal gland and all the accessory structure of the eye. There is extrinsic eye muscles, means the muscles which is attached outside the eyeball. So eye has two kinds of muscles. Keep in mind, eye has extrinsic muscles and intrinsic muscles. Okay. So extrinsic muscles are outside and they are voluntary muscles. So you can move your, like if you contract those muscles, that moves your eyeball. An intrinsic muscle is inside the eye, and intrinsic muscle function is to regulate the size of your pupil and to regulate this thickness of your lens so that you can focus any image on the retina. Okay, so that is the extrinsic and intrinsic muscle.
Are the intrinsic muscles, you said they are involuntary, and extrinsic is voluntary?
Yeah. Okay, here. So here is the eyeball, and this is the frontal section, the sagittal section of the eyeball. So the muscles. So you see there are these muscles. The muscle originate from common tendinous ring from the orbital cavity, means your bone in the orbit, and then it inserts on the sclera, the white layer of the eyeball. Okay, so there is superior oblique muscles, inferior oblique muscles, superior rectus muscle, inferior rectus muscle, lateral rectus muscle, and medial rectus muscle. Okay, so there is right lateral, left lateral, everything right and left in each eye. Okay, these muscles moves your eyeball. If you see from the front view, interior view of the right, you see here, these are the muscles and the direction, arrow showing if they contract, which way your eyes will go. Okay, now the eyeball, the tunics of the eye, which is the most important part of the eye. So eye has 3 tunics. If you take eyeball, and you're going to see at the end in the dissection, if you take out the eyeball and cut it and see from outside in, you're going to see 3 layers. Okay, so those 3 layers, the outermost layer A is called fibrous layer. The middle layer is B, that is called vascular layer. And C, the innermost layer is the neuronal layer or sensory neuronal layer, sensory layer. Okay. So most external layer to the eye is the fibrous layer. You see here, most external layer of the eyeball composed of 2 regions of the connective tissue. And we're gonna talk these all here. Without diagram, you're not gonna understand. So I'm gonna show you diagram. Look at this diagram and go over each terminology one time. So when I explain, you understand. Don't ignore. I'm gonna pick someone from your group and all of a sudden I'm gonna ask, and don't say then, oh, I don't know where it is. It is in this diagram. You have to go over one time. Just go over these all terminology. I have posted in Canvas these all terminology for both lab and lecture. Pretty much they are common. Sclera, choroid, retina, macula lutea, fovea centralis, posterior pole, optic nerve, central artery and vein of the retina, optic disc, optic or blind spot. Posterior segment contains vitreous humor, sclerovenous sinus, lens. Interior segments contains aqueous humor, interior pole, pupil, iris, cornea, ciliary granule, ciliary body, and ora serrata. So this is subject's right eye, and frontal section of the eye is removed, and you are looking at the posterior half of the eye. This is the posterior part of eyeball, and this is anterior pole of eyeball. Okay, so as I said earlier, eye has 3 tunics. Tunics means layers. And they are, what are they? I just wrote earlier. Somebody please.
Layer.
Okay, so the outermost layer is fibrous layer, and the fibrous layer are made up of dense regular connective tissue, and that is called sclera. The fibrous layer has 3 parts: sclera, cornea, and it is the part of optic nerve here outside. The next layer is the dark layer. We call it vascular because it is full of blood vessels and a lot of melanocytes. That's why they are dark. And the vascular layer is called choroid. You see, choroid has, the middle layer has certain parts. It is choroid, it is ciliary body, it is iris. So iris, ciliary body, and choroid are the part of vascular layer or middle layer. Then innermost layer is sensory neural or neural layer, because they are made up of usually the nervous type of cells, and that is this yellow posterior layer. This is the only layer which has the receptors for the vision, that is rods and cones. Okay, so imagine your eyes. Let's talk about again. So this is the posterior pole here, and there is the sclera. Sclera start from here, comes anterior. Sclera is opaque. That means light cannot pass through the sclera. But when a sclera comes anteriorly, this area, sclera becomes transparent. That means light can enter through the sclera here. And that transparent region from here to here, this region, is called cornea of your eyeball. And this is the most anterior portion of your eye. There are several layers of the cornea, and the outermost layer is made up of epithelium, which contains receptors. That's why when some like dirt or anything comes into your eyes, they irritate your cornea outer surface, and you have blinking reflex to remove it. Okay, so that is the
Professor,
uh-huh.
What is the layer that they remove when you do LASIK surgery?
Yeah, so a LASIK surgery will burn the cornea layer. Like if you have irregular surface or some opacity, they come and they just scrap them and make them smooth with very tiny surgical knives. So this is cornea layer.
Thank you.
Okay. That's called LASIK. They use laser and they remove the surface. And when you donate your eye, you don't donate your whole eyeball. You donate only your cornea. So you have, if you are a eye donor, they will not come after your death and take out, scoop out your whole eye. They will take only the cornea. Okay. The junction of sclera and cornea, you can see there is scleral venous sinus. This area is, this junction is called limbus, and there is a scleral venous sinus. This is the area which makes aqueous humor here and put into in this anterior segment. We'll talk about that later. Next, second layer is choroid, which is the vascular layer. When it comes anteriorly, it modifies and then it becomes ciliary body. Ciliary body contains ciliary muscles. Ciliary muscles are extrinsic muscles or intrinsic. Is it inside the eye or outside the eye?
Inside?
Inside. That's why it is intrinsic muscles of the eye, and they are involuntary muscles. These ciliary body contains ciliary muscles, and it is attached to the lens through the ciliary zonule, which regulate the size of this lens, and that's how you focus the image on your retina to see the image clearly. Most anterior part of the choroid is the iris, which cover the lens, and iris has also intrinsic muscles in this area here. And there are two kinds of muscle. The inner circular muscle here, and outer here, radial muscles. And contraction and relaxation of these muscles can increase the size of hole in the cornea. And this hole inside or defect inside the cornea is called pupil. So pupil is not a structure, it is a hole, a defect in the choroid. Are you following me?
Professor, can you go over the two types of muscles again, please?
Radial muscles and constrictor muscles, or we call them circular muscle, radial and circular muscles. And they are also intrinsic muscles. Okay, so that is the choroid. And then you have crystalline lens. This lens is very good media to pass the light and bend the light so that it falls on the retina. Now you have retina. The retina then forms the optic nerve, and optic nerve, core of the optic nerve is the passage of central artery and vein of the retina. So all the artery and vein comes, branches off, and you see these small artery and veins are running in the wall of the eye. Now retina has some special feature. So whole retina contains two kinds of receptors, rods and cones, which we see later. Most of the receptors are rods, like more than 90% are rods. Very few are cones. There are some area where there is no receptor in the eye, like here, no rods, no cones. And that area is called optic disc. Because there is no receptor, you will not see anything from this area. And that's why this area is called blind spot. Here is another pit, macula, and fovea centralis. This is the only area inside the eye where you have a lot of cones. Otherwise, most of the eye has mainly rods and very little cones. This is the area where in macula, you have mostly cones, and center of the macula, fovea centralis, has only cones. There is no rods. So when you have to see anything clearly, the image must fall here. Okay. This is in this diagram, I'm talking like my 7, 8, 9, 10 slides. So you have to pay attention. These all are explained in coming slides, and I'm just explaining. Now, let's go about these 3 layers. Let's go inside the cavity of the eye. Is this cavity of the eye empty? No, it is not. So the area behind the lens, this whole area, we call them posterior segment. And to hold this retina, the choroid, everything attached to the sclera and in the wall of the orbit, this posterior segment is filled with a jelly-like, very vitreous liquid, and we call them vitreous humor. Okay, So this is posterior segment, this whole area. In front of lens from here up to the hair, up to the posterior surface of cornea. This whole area is anterior segment. And this area also contains liquid produced by scleral venous blood vessels here. And this fluid is very watery. That's why we call them aqueous humor. Okay. This aqueous humor provide nutrition, maintains waste product of the eye, anterior part of the eye. Okay. Now, one more thing here. You see in the anterior segment, so what is the boundary of anterior segment? From lens to, someone please.
Ciliary zonules.
Anterior segment is from lens to cornea, this all area from here up to the hair. From here up to here, or from here up to here, or from here up to here. See, this all area. Now this area, this area can be divided into, anterior segment can be divided into two regions. This is small dark area here, in front of lens, behind the iris, and in front of iris and behind the cornea. And these two areas is called chambers because they are chambers. So anterior segment can be divided into posterior chamber and interior chamber. Okay. Sometimes we have increased fluid, aqueous humor, and increase the pressure in the eyeball. That condition is called glaucoma, increase intraocular pressure. We are born with vitreous humor. You don't produce this every day. And if you have lack of vitreous humor, then in that case, there is chance of retinal detachment, and that causes complete blindness. But aqueous humor you produce every day, it comes here and then absorbed again back. So produced here and then absorbed here, okay? Produced by this area in the ciliary body. Then it comes here through this area and come here and absorbed here, okay? So most important part here, anterior segment, posterior segment. Anterior segment had two chambers, anterior chamber and posterior chamber. You guys need to know that, okay? Okay, so vascular layer, we talked about it. Here you go. Flow of aqueous humor. You see, in the ciliary body, you have ciliary muscles. What is the function of ciliary muscle, by the way, we talked about earlier?
To increase and decrease the size of the iris.
Ciliary muscles. Ciliary muscle is attached to ciliary process and then to the ciliary zonule and then to the?
To focus the lens.
Yes, it regulates the size of the lens. Your pupillary muscles are here. Pupillary dilator, pupillary constrictor, which is called circular, and radial muscles are here. That regulate the size of the pupil. So this muscle regulate the size of your lens. So you see, the aqueous humor is produced here from the posterior chamber, then it goes through the pupil, anterior chamber, and absorb here in the scleral venous sinus, and then get into your heart. Okay, let's talk about the retina. Retina is the diffuse tunic composed of two layers, pigmented layer, single layer of melanocyte, and neuronal layer. That is the layers of nervous tissue and contains three main types of neurons: photoreceptor cell, bipolar cells, and ganglion cell. So let's see in the diagram. This is the eye. This is the posterior pole of the eye. This is anterior pole of the eye. And we are eye, and we are here. This is the inner side of your eye, retina, and this is outer side. Okay. So here is inner and here is outer side. So let's see if light falls here, cornea, pupil, lens, and then let's see fall here. Then where it falls first? Pathway of light. It falls on the this way. So what are the layers of the retina? Retina has 10 layers of the cells, but we're not going to go in detail here. So the outermost here, if I go from outside here, here will be sclera, then choroid, and then retina. Okay. So retina has first layer. This is called pigmented layer of retina. It contains a lot of melanocytes. And in the pigmented layer, you have rods and cones. So this is like regular retina layer. Can you compare the rods and cone photoreceptors? Which one is more, rods or cone?
Rods.
Yeah.
Rods.
So most of the receptors in the eye are rods. Rods are for black and white vision. That means they are active only during the nighttime. Whereas cones are responsible for color vision or daylight vision or bright vision. Okay. So after pigmented epithelium histology, you have photoreceptor layer. Then you have bipolar cells. You see, two poles. As I remember the structural classification of neuron, bipolar cells. Then ganglion cells. Inside the ganglion, there is some another cells. You see, amacrine cells. And the axons of ganglion cells. Axons of ganglion cells, these all actions pull from there and comes posterior side. And that is called the optic nerve. So these all actions of ganglion cells. Actions are the long extension of the ganglion cells, yes? They make a bundle and exit through the posterior pole of the eye and becomes the optic nerve.
Professor, I have a quick question.
Uh-huh.
So for someone who's colorblind, is that because they lack cone cells?
Yeah, there are three kinds of cone cells, like red, green, and blue. So what kind of you have lost based on that, or defect in the receptor, you will have color blindness. Yes.
Can you
Okay.
go
So
to the previous slide, or actually, I can hold my question until after class.
okay. Thank you. Okay. Photoreceptors are two main types: rod cells and cone cells. Rods are more sensitive to light, allow vision in dim light. Cone cell operate best bright light, enable light. High bright light enable high acuity color vision. So when you, let's say, when you want to see certain things very clearly, then the focus must activate your cone cells. And that's why we manipulate our head when we are looking into at a dot. So if you have a small dot on the wall and you want to focus on that dot, the image from that dot must fall on your fovea centralis, where there is a lot of cones. Okay. These all receptors are considered like modified neurons. Okay. Some regional specialization of the retina again. So those are I have talked about. Ora serrata, macula lutea, fovea centralis, and optic disc. Ora serrata retinae are neural layer ends at the posterior margin of the ciliary body. Pigmented layer covers ciliary body and posterior surface of the iris. So let's go and talk about this ora serrata. You see here, this is the choroid layer, and there is the retinal layer. So retinal layer is ending here, and choroid and retinal layer are making contact with each other, like the interdigitating, and that area is called, that margin is called ora serrata. Okay. So internal chambers and fluid, we talked about it. The lens and ciliary zonules divide the eye. Posterior segment or posterior cavity filled with vitreous humor. They are clear, jelly-like substance, transmit light, and support the posterior surface of the lens, helps maintain intraocular pressure. Whereas anterior segment divide into anterior and posterior chambers. Anterior chamber is between cornea and the iris, or iris and cornea. Posterior is between iris and lens. Filled with aqueous humor, they're renewed continuously, formed as a blood filtrate, and supplies nutrition to the lens and cornea. So anterior chamber nutrition is supplied by aqueous humor. And this is the flow of the so let's go, and let's see how we see when we see an object. So let's see, when I raise my finger and you guys looking into this finger, what happens? Image, light falls on my finger, and then image from my finger, the light reflects. Some of the light is absorbed by my fingers, some of reflecting. And that light, like when we see object, we are not seeing basically object. We are seeing only light. Our brain process it, okay? So reflection of light then, let's see arrow here. So when the light arrow comes here, when it hits the cornea, cornea is different density compared to air. So when it changes the density, then it bends from the image. Let's see, this is the image, and you are looking at. So it comes then, it bends, and then through the pupil, passes down here. Let's see, if you are in dark room, then your pupil size increase so that more light will enter. If you are in very bright light room, then what happens? You constrict your pupil. When somebody is taking drugs like heroin, morphine, what happens? Their pupil is like very constricted. So pinpoint constricted pupil. When there are certain drugs which dilates your pupil. When pupil is dead, somebody is dead, we check the pupil, whether pupil is dilated or not, and you see wide open pupil dilated. So one light passes here, it enters through the pupil and then hits again the lens. Lens is even thicker than the cornea. And based on where object is coming from, it is from 20 feet or away from 20 feet. Based on that, ciliary muscle would adjust the thickness or concavity. Like this is the lens. You can increase or decrease the concavity of the convexity of the lens and increase the surface. And then ultimately you adjust the lens in such a way that image, then this image will pass here and ultimately falls on the retina. If the image falls behind the retina or in front of the retina, then you have visual defect. Okay. Once image falls here, we see the image inverted, like upside down. The receptors then produce action potential, and in the retina, action potential is hyperpolarization. You don't need to know, this is physiology. And that impulse then through the axons of ganglion cells becomes the optic nerve. Then optic nerve come here, and in the middle, it cross in the optic chiasm. Half of the fibers cross, and then it goes to the occipital lobe. And in the occipital lobe, you see the light, and then from there, visual association area, you recognize the image, size, shape, color, and then you understand it. Okay. Okay. Okay, visual pathway now. So as I said earlier, let's see, this is your eyes. Just think this is your eyes. This is your right eye. This is your left eye. And here you are looking into this object. So you can see with either eye, you can close left or right, and you can see this object. Okay. So image from this object, you see it falls on the retina, and retina has two parts. The lateral part, which is called temporal retina, and this is medial part, which is called nasal retina, because it is towards the nose. So the axons from lateral part is coming here, and nearby the hypothalamus in the diencephalon. The nasal part of the optic nerve, which is towards the medial part from both sides, is coming and crossing. So from the right eye, it is going to the left side, and then some of them synapse in the superior colliculi. Superior colliculi is the part of which part of the brain?
The hypothalamus.
Nope.
Mid,
Midbrain. Yes, it
Brain.
is midbrain. Midbrain. And then lateral geniculate body here in the thalamus. And then you comes to the occipital lobe. But the temporal fiber from both sides, you see, is going to the same side of the brain. They do not cross. And this is called optic chiasm. So pathway of the neural pathway is retina, optic nerve, optic chiasm, optic tract, and then superior inferior colliculus, lateral geniculate nucleus, optic radiation, and occipital lobe. Okay. This is the dissection of the eye, and this is a cow eye, and you can see this is the eyeball with cornea. Cornea is clear, but after it's dead, the cornea oxidized and becomes dark. This is the optic nerve here, and you see a lot of muscles attached to it, excess fat and muscles, and then sclera. This is the anterior segment, anterior half filled with vitreous humor, and then posterior segment, you see gooey, very like fat-like substance. That is the retina, and it is the back view of anterior pole. So anterior pole, back view. When you see from the back, you see the lens and the ciliary body, ciliary zonula attached, and you have cornea removed, clear here, and the posterior segment, you have retina. All the lists of the organ and video, you can watch this video here, dissection video. Okay. That's all for today, guys. If you have any question.
I have a question.